So, welcome to the next module of biochemistry. It is amino acids and proteins, and these are the topics which we are supposed to discuss in this particular module. Okay, first we can see what are proteins. So, proteins are the most abundant biological macromolecules occurring in all the cells and all parts of cells, and they are actually made up of amino acids that are covalently linked. What is the major role? Okay, you have been learning about proteins from your lower classes whenever you have been studying about the diet, food, and all. So, what is the role of protein in the body? Okay, it has, we can divide it in the structural role and diverse role. In the structural role, we can specify a few examples like collagen and elastin, which are proteins that are found in bone matrix, then vascular system and other organs. There are alpha keratin, which are present in the epidermal tissue. So they are actually have an important role in this as a structural framework. Okay, so it provides what you call structural component to these what you call areas: bone matrix, vascular system, and epidermal tissues, etc. Now, what are the diverse roles played by proteins? They are enzymes. Enzymes. There are actually majority of enzymes are proteins. There are non-proteinaceous enzymes, obviously. Hormones, proteinaceous hormones are there. Blood clotting factors. All the blood clotting factors are proteins. Immunoglobulins, or what we call as the antibodies, they are protein in nature. Membrane receptors. Membrane receptors. The receptors. For various factors which are found on the plasma membrane of cells. They are proteinaceous in nature. The storage proteins, obviously, they are proteins, and besides, they also have a role in genetic control, muscle contraction, respiration, and plenty other. Right. So, as we have already mentioned, proteins are formed of amino acids, which are linked by covalent linkage, isn't it? So, typically, amino acids are monomers that make up the polymer proteins. Okay, and these monomeric units are connected by covalent bond. And specific type of covalent bond found in between amino acids, they are known as peptide bond. Okay, so now what is amino acid? Amino acid is a organic compound that contain both functional groups, that is basic as well as acidic, basic amino group and acidic carboxyl group, and that is why the name amino acid. Okay, so it contains two different functional groups, okay, in the same molecule. Now there are about 20 different amino acids that are commonly found in proteins, that make up the proteins. And the first to be discovered was asparagine in 1806, and in 1938, 309 came into being, and this forms the 20 amino acids. Selenocysteine is one of the recent most discovered amino acid. Okay, all the amino acids have common names like asparagine, which has actually taken its origin from asparagus, which because the discovery was made from asparagus, extraction from asparagus. Glutamate from gluten, wheat gluten. Tyrosine from tyros, which refer to cheese in Greek. Okay, and it was first isolated from cheese. That is why the name tyrosine. Glycine, because it is sweet, and the name came glycine. Okay, so likewise, each amino acid, if we look, the naming do have a history behind it. Fine. Now we can see the basic structure of amino acid. Amino acid, this is the structure. Okay, so there is a central carbon, and this carbon is referred as alpha carbon because the functional group is attached to this carbon, isn't it? So this is the alpha carbon. Okay, it is mentioned over here. Now the alpha carbon is to the alpha carbon, you can see there is a carboxyl group attached. Okay, the whole thing is the carboxyl group. There is a hydrogen atom, there is a amino group, and there is a side chain. Side chain can be simple hydrogen, and it can range up to like branch chains, or what you call aromatic benzenoid compounds, amino compounds, etc. Okay, so R, it varies from one amino acid to the other, and that is what, depending upon the R, the property of the amino acid also changes. You can see the rest of the molecules, they are the same. Okay, the carboxyl group, it is found found attached to the alpha carbon in all the amino acids. Likewise, the amino group, it is also attached to the alpha carbon and the hydrogen. So these remain the same in all the amino acids. What changes? It is the side chain. Okay, since it is alpha carbon which binds to these functional groups, we call it as an alpha amino acid. Okay, now you can see this is a general structure. Okay, and what happens is in natural conditions, in biological system, always the amino acid exists in ionic form. Okay, in this form. Okay, it is COO minus and NH3 plus. Okay, so you can see it is positive and negative in the same molecule, and hence it is what you call neutral charge. Okay, and we can say it is dipolar. Right. So this is the structure of amino acid. Okay. Now, when we speak about amino acids, as we have already mentioned, there are 20 amino acids which take part in the formation of proteins, and they are all referred as alpha amino acids because the amino group and the carboxyl group, it is attached to the carbon, and that carbon is referred as the alpha carbon. Okay, it consists of the central carbon, which is known as the alpha carbon, and linked to the alpha carbon, we have the amino group, carboxyl group, hydrogen atom, and side chain. And it is this R group or the side chain which varies from one amino acid to the other. Okay, and amino acids differ from each other in their side chains, which vary in structure, size, electric charge, and this influences the solubility of the amino acids in water. For example, if the R group was contains hydrophobic compound, obviously the whole amino acid becomes hydrophobic in nature. If the R group is more hydrophilic, obviously the whole amino acid becomes hydrophilic. Okay, so the properties, the chemical or the physical properties of the amino acid depends upon the R group. Okay, now alpha amino acid, it is chiral in nature, that is it is asymmetric. We can see, right, this carbon, it is attached to on one side carboxyl, the hydrogen, amino, and a side chain. So these four groups are completely different. Okay, so the four valencies of the central carbon, it is filled with four different groups, and hence this alpha carbon is asymmetric in nature, or it is chiral in nature. So what happens, this particular molecule becomes optically active. Okay, except in one case, when as already mentioned, the R chain here, the R can range from simple hydrogen to, it may go up to like benzenoid compounds. Okay, so when it is hydrogen, what happens, the two valencies are filled with the same group, isn't it? So there is no asymmetric chirality here. Okay, but otherwise, except for that particular case, in all the amino acids are optically active due to the presence of asymmetric alpha carbon. Okay, now because of this alpha carbon, there are two optical isomeric forms, and they are L isomer and D isomer, and we can see that only the L amino acids are constants of proteins. Okay, I hope you remember when we discussed about carbohydrates, we saw that it is the D glucose or the D isomers which take part in the formation of oligosaccharides and polysaccharides, and it is the D isomeric forms which are more biologically active. Okay, and here you can see the amino acids, they are L amino acids, they are that take part in the formation of proteins. Okay, so this is the structure of the isomers, and you can see here they are optically active, and they are this is the serine molecule. Okay, and here what is it, L serine and D serine, what is the difference? The difference is with respect to the spatial arrangement of these molecules, isn't it? Okay, on either side. Fine, and we can say that L serine is biologically active, L isomer is biologically active. Okay, so these are known as stereoisomers, because stereoisomers, what does it mean? Molecular formula is the same, only the spatial arrangement of the functional groups, it is different, isn't it? So here you can see the arrangement is different. Fine. So these are the stereoisomers. Okay, right. And apart from these, we can see that amino acids also do have special other properties because of its specific nature. Okay, amino acids in solution at neutral pH exist predominantly as dipolar ions. Okay, so what happens is when amino acids are in aqueous medium, amino acids, they usually remain in this form. Okay, having two ions on the two, like positive and negative charge on the same molecule, and this condition is known as a Zwitter ion. Okay, Zwitter ion, or it is otherwise the dipolar ion. Okay, so amino acids usually they remain as Zwitterionic in solution form. Okay, when in solution. In dipolar nature, the amino group is protonated, that is NH3 plus, and carboxyl group is deprotonated, that is COO minus. So the ionization state of an amino acid varies with pH. Okay, that is yet another case. That is when it is in more acidic condition, what happens? More acidic means it contains more of H plus, isn't it? And what happens when there is more H plus? When there is more H plus, it can bind with COO minus, isn't it? Okay, so what happens at that time? So we can see here. So just imagine this is amino acid, and this amino acid, it is placed in a highly acidic medium. Highly acidic medium means it contains more amount of hydrogen ions, free hydrogen ions, and these free hydrogen ions, that is H plus ions, it will bind with COO minus. Okay, so it becomes COOH, but NH3 plus remains the same. So what is the charge? Net charge is positive. Okay, on the other hand, when we go towards a highly alkaline condition or highly basic condition, what happens? Basic condition contains less amount of hydrogen ions. Okay, so in high basic condition, the NH3 plus, it will give away the hydrogen and it becomes NH2. Okay, so what happens to this particular amino acid? It will have COO minus and NH2 here. Okay, so what is the net charge? Net charge becomes negative. Okay, so the charge of the amino acid also changes uh, or varies with the pH. Okay, so in acidic solution, the amino group is protonated, NH3 plus itself, but the carboxyl group is not dissociated. Instead, it will become COOH. As the pH is raised, that is, it becomes more and more basic, the carboxylic acid is the first group to give up a proton. Okay, and then obviously the ionic ionization state will change. Okay, so this is how it is represented. So we have, this one is the Zwitter ion form. You can see the same molecule is having positive and negative charge. It is a dipolar condition, and this is Zwitter ionic form. This is a Zwitter ionic form. Okay, what happens? We can see when more amount of hydrogen ions, when hydrogen ions is introduced. What is hydrogen ions introduction? It is becoming more and more acidic, isn't it? So when more amount of hydrogen ions it is introduced, what happens? The COO minus takes up the hydrogen. Okay, and the net charge, it becomes is positive charge. Okay, so both groups are protonated. Okay, so what is the charge? It is positively ionized. Okay, on the other hand, when you can see the arrow on the top. Okay, what happens here? When hydrogen ions it is removed, so it is a basic condition. Okay, what happens then? What happens here is actually that the NH3 plus, it gives off the hydrogen. Okay, CO minus remains the same like this. Okay, so when in such a condition, when it is like it becomes more and more basic, what happens is the COH, it gives away the hydrogen ions readily. Okay, both groups become deprotonated. Okay, so it becomes negatively charged. Okay, so the ionization state, it changes with the change in the pH. Okay, so this is what it happens. Now, it is about the classification, and the classification, there are different types of classifications based on different criteria, and that we'll be learning it in the next presentation. Okay, fine. I hope it is clear. Right. Thank you.